Hello everyone, and welcome to the webinar today on optimizing cell therapy manufacturing through custom single-use solutions, brought to you by RegWinNet. My name is Elena Conroy, and I will be your host for today's event. I am the commissioning editor for Future Science Group, publisher of the journal Regenerative Medicine. We're delighted to have Natasha Lundgren from Saint-Gobain presenting for us today. So a little bit about Natasha. So Natasha Lundgren is the product manager for cell therapy within Saint-Gobain's Bioprocessing Solutions business unit. She joined Saint-Gobain in 2012 and previously served as a process development engineer within the CertainT division and a research engineer within the Performance Plastics division. Natasha received her BS in chemical engineering from the Pennsylvania State University, where she supplemented her studies through research internships in bioengineering and immunology. So before we begin the presentation today, we have some quick poll questions that we would be very grateful if you could answer. So for each poll, please check the option or options that apply to you, and then you can just click the submit button. So the first question for today is, are you aware of Saint-Gobain's single-use product portfolio? And the answers are yes or no. Thank you very much. So it seems like it's 50% for both at the moment. So the second question is, what life science sector best describes your current work? Small molecules, biopharmaceutical, cell therapy, or other? Okay, so it looks like most of you are working in cell therapy. Right, so the final question for now is, are you currently involved in the manufacturing of autologous cell therapies, allogeneic cell therapies, both or neither? Thank you very much. So that was the last question. Thank you very much for taking the time to answer our poll questions. So we invite you to submit any questions you may have for our presenters throughout the whole webinar using the Q&A tool to the right-hand side of your screen. We will pose your questions to Natasha in a live Q&A session after the presentation, and then follow up with any outstanding questions offline. So thank you everyone for joining us today, and Natasha, I'll now hand over to you for your presentation.
Thank you, Elena, and thank you everyone for joining today. So today's discussion is about optimizing cell therapy manufacturing through custom single-use solutions. And I'm happy to have gotten the poll results at the beginning, so it's nice to see that there are some that are new to the Saint-Gobain portfolio and some that are familiar. So I think that this presentation should be informative for both parties as it discloses more on some custom solutions that we've done, but also overviews some of our interesting products for cell therapy. So just to orient everyone to the agenda, so I'll give a quick introduction to Saint-Gobain for those of you on the phone that are not aware of the company. Then the bulk of the presentation will be spent in looking at methods and examples of designing custom closed solutions for cell therapy process steps through a variety of case studies. And then we'll end with looking at a zoom on a very complex case study, which is the Argos case study. So Saint-Gobain is a very large company. It's operated worldwide in over 65 countries with almost 200,000 employees, and is a 40 billion euro company, almost 40 billion euro. And it was named one of the world's top 100 innovators. We're actually a very old company as well. Last year we celebrated our 350th year anniversary. We were started in 1665, actually by Louis XIV, where he commissioned for starting glass manufacturing. And through the years we've acquired different markets in going into construction as well as in plastics. But at the heart and root of Saint-Gobain, we're truly a material science and a manufacturing company. So funny enough, our first product is still in use. 50% of the mirrors in the Palace of Versailles Hall of Mirrors are the original glass that was made by Saint-Gobain. So zooming in a little bit onto life sciences, and this is the portion that we sit in today. So Saint-Gobain, as I disclosed a little bit, is in multiple markets and multiple industries. But the industry I'm going to talk about today is the life science sector. And this is really where our solutions are focused on fluid transfer, whether that be for medical devices, analytical equipment, bioprocessing, and of course biotech. So our product offering specifically for bioprocessing solutions spans a wide variety of products. Fluoropolymer bags is one of our leading products in the cell therapy industry for both cell culture and cryopreservation, given the inherent properties of fluoropolymers. But we also have bioprocess containers, which are great for media transfer and storage. We have a wide variety of connectors to make aseptic connections. We offer a wide variety of fluid assemblies. Filters, we acquired a company last year that allows us to make custom filters, which really fits nicely into our ability to make custom solutions. And of course, many of you on the phone may know us for our tubing, so C-Flex and Tygon are our leading brands in the bioprocess and biotech industries. So really with all these products, at the root of it is manufacturing capabilities, and that comes from both material science as well as from process and manufacturing. So we work with a wide range of materials with a focus on high-performance plastics. So this could be silicones, fluoropolymers, as I mentioned, other high-temperature and engineering plastics, and thermoplastics. And really, the point of it is choosing the right material for the application and really understanding the application, understanding what the material has to see, whether it be sterilization effects or mechanical properties or what manufacturing process it needs to go to. So this is really the core of Saint-Gobain, is this material science and material selection aspect. And on top of that, the ability to process material. So after you choose the material, then understanding how to process that. And we also have a wide range of process technologies going from extrusion to injection molding, film lamination, of course bag fabrication, and the ultimate assembly of fluid transfer assemblies and solutions. As I alluded to before, Saint-Gobain is a global company, and that's no different for the life science sector. The life science sector also operates on a global footprint. We're operated on almost all of the continents: Europe, Asia, South America, and North America. Gaithersburg, Maryland is actually our center of excellence for biotech and cell therapy. It's where we manufacture a lot of our products for cell therapy, including our fluoropolymer bags. But it is not the exclusive place for manufacturing in the life science space. We have many capabilities that span across the continents with thermoplastic extrusion, molding, silicone extrusion and molding, of course the fluoropolymer extrusion of the film, the bag manufacturing, polyethylene bag manufacturing, assembly, and our filter manufacturing in Hangzhou. So really this is getting into the meat of the presentation and really understanding how Saint-Gobain works in developing custom solutions. And it is a very interactive process, as hopefully you'll see throughout the presentation and the case studies that we give. So it usually starts with a meeting with the customer, typically a WebEx to work through the design, really be able to provide a solid engineering drawing for the customer to sign off on and agree with. Then we typically provide a sample for testing. Usually a drawing does not always translate to exactly how it's going to look and feel, so it's important that the customer can get something in their hands, put it in their system, understand if there's any changes that are required, and then and only then do we look at delivering the final custom product. And this is really the approach we've taken with the next case studies that I will disclose. Okay, so today's is going to focus, I know some of you are not in the autologous space, but for the purposes of just orienting everyone, a lot of the examples I'll go through today were focused on autologous, but of course can be expanded out to allogeneic. Of course, the challenge with allogeneic is the scale up versus the scale out, but that's certainly a challenge that can be discussed. So the case studies today are oriented around each step of the process chain for autologous cell therapy, starting with cell separation and sorting. So this is an example where there was multi-parameter cell sorting happening using flow cytometry to be able to sort the cells on more than one surface marker. And so using the flow cytometer, they were using a reusable sheath tank, which caused a lot of effort to be able to clean and also to validate the fact that it was clean, and ultimately posed a higher risk for contamination. So we worked with the customer to build a bag and a fluidic set that could actually be integrated into the sheath tank to transfer the fluid to the flow cytometer in a closed and sterile manner, and for it to be single use. This is the concept of the design. The sheath fluid would come into the sheath bag that was fitted into the sheath tank, and then be able to get the fluid out and processed into the flow cytometer. This is what the design looked like. As you can see, there's a tubing assembly that goes into the bag. That's how the sheath fluid would go in. And then you see at the bottom there's a rigid FEP tubing, and that was the ability to completely drain the bag and also not collapse under pressure. So this goes back to understanding material selection and looking at the criteria that the product has to perform in. And so this is where tubing had to be identified that could hold up to the pressures of the application. And that story remains true with the connections that we had to make to the flow cytometer, looking at pretty sophisticated connections, including metallic components, in order to make sure that it could hold the vacuum under the application of the flow cytometer. This is a very interesting design as well, with the bag having a very specific shape to it, again, to encourage the ability to drain all of the sheath fluid into the flow cytometer. This is the solution we developed for PCT that was supportive of their phase 2 clinical manufacturing of a Treg therapy. So moving on to cell culture. So many of you that might be working with a cell type like T cells that are expanding typically understand that there's a seed train involved, and that may require a multi-bag system. So the challenge, of course, is when you do a multi-bag expansion, it often requires open transfer steps, whether that has to be performed in a biosafety cabinet or you're using pipettes. But essentially, the open transfer obviously alludes to potential risk for contamination. So solutions, we looked at a system comprising both a custom fluid transfer set and a series of bags that would allow for expansion to occur in a closed system. This is the concept. So you'd have your media, your cells, your cytokines. You'd seed into 2 small culture bags, and then when they become confluent, then you would be able to seed them into a larger culture bag. This is what the design looked like. So you have a syringe ability to add and seed into 2 smaller bags, and of course all this tubing, depending on the application, could be pumpable, it could be sterile dockable, it could be weldable. So those are all considerations that, based on the customer's requirements, you'd have to consider. In this case, it was sterile dockable to allow for the feeding of the cells and the disconnect. These would then be put into the incubator, and when they reached confluence, they would be pooled and seeded into the larger bag. And here again, you have a very specific shape on the larger bag, and again, that's to enhance both filling as well as draining to make sure you can get all the contents out of the bag. This is a system we designed for City of Hope, again used for the expansion of T cells in a clinical trial for non-Hodgkin's lymphoma, and there is a reference here that discusses the use of this system in their process. Moving on to cell harvest. So a lot of the times our customers are using magnetic beads, and the magnetic beads, of course, can be used at the beginning of the process for the separation of the cells, but they're also used during the process, especially again for T cells where you're doing an activation step. And so at the end of this activation step, if you've removed the beads from the cells, you now have a mixture of beads and cells after culture. And so the challenge is if you need to separate those beads from the bead cell mixture, it can often be limited to open systems. So we looked at doing a bag and tubing set, including a bag with a very unique flow path that would allow for bead removal and cell harvest to happen in a completely closed system. This is what the concept looked like. So you'd have your culture bag with your beads and your cells in a mixture, and this is assuming that the beads have already been separated from the cells, but they still remain in the solution. And so typically you would run over a magnet. Now what we looked at is doing a very specific design that would allow for multiple passes over the magnet. So this is more of like a snake serpentine shape. And that would, between the flow and also the multiple passes, we saw that that offered really nice separation of the beads from the cells. So with the magnet on, you could flow the solution through and collect the cells directly for collection for the next step or even for freezing. And then you could turn the magnet off and actually collect the beads depending on if you were reusing them or just disposing of them. So this is what the concept looks like. So again, you have a very nice design for the bag to enable draining. You have multiple passes through the serpentine pathway, and again, this is all customizable in terms of how many loops you'd want, what the dimensions of this are. And then you have the ability to go right into freezing or into a waste container for the beads. And this is also a design that was supportive of PCT's Phase 2 clinical manufacturing. I also have an image of this, which is nice to see with the fluid in it. Here we go. So here you see that the flow path given the fluid, so it's that nice serpentine path, and then the fluid or the cells could go to the freezing bag, which is shown in the orange, or the beads could go to the waste. Okay, cell concentration. So cell concentration, one of the gold standard ways of concentrating cells right now in the industry is centrifugation. But unfortunately, centrifugation is often, again, limited to open systems and transfer steps, including pipetting, especially when using conical tubes. And this presents, again, a high risk for contamination. And at this point, you're dealing with the final product. So the risk of contamination is even higher due to the fact that you've brought this product all the way through the process. So we looked at creating a disposable set that included a conical tube, but with a custom cap that allowed for cell concentration to be completed in a closed system. This is the concept. It's a little more involved than some of the other examples, but essentially you'd have your culture bag. You'd be able to sterile dock onto a custom cap, remove all the tubing, spin down, remove the supernatant, again by sterile docking on tubing, resuspend by sterile docking on a fresh media bag, and then be able to put the whole solution into either a cryopreservation bag or into a bag for the next step. So again, this is the drawing that it looks like. So there's some sterile connections that need to happen, but essentially what you have here is that you have the cell culture mixture being able to be put into the tubing, right? So it's put into the conical tube. And as you see the zoom, you have the conical tube cap, you have the connection for the tubing, and you also have a vent, and this allows for the filling of the conical tube. You could then remove the supernatant. That could go to a waste or a barrier bag. You could resuspend with the media, which actually has an inline filter. So you can sterile fill the media into the conical tube. And this could be either a washing solution, or it could be the DMSO, or it could be cell culture media, depending on what stage of the process you're in. Once you resuspend, you could then move to the next process, whether that be freezing or a separate holding bag. And the last example in this part of the talk is for final fill cryopreservation. So aliquoting for final fill and cryopreservation. So filling final product, if you're doing it into small aliquots, whether it be for single doses or for sampling as well, it can be time consuming and require, again, open transfer steps for individually filling either cryo vials or small bags. We looked at creating a disposable set that included an overmolded assembly, which allowed for a smooth interbore transfer connected to small bags that allowed for final filling to occur in one closed system. This drawing is a little maybe difficult to follow as well, but basically you would come with your bag that had the concentrated cells from the previous step. You do your formulation with cryomedia and end up with your cryopreserved cells, concentrated cells in a cryopreservation media. Those could then go and fill to small cryo bags. And again, this is customizable in terms of how many bags are on the line. This is what it looks like. You have your bags. You have a syringe to be able to add in any cryoprotectant media to get to your concentrated cells in cryoprotectant. And again, this is where material selection is important. DMSO, DMF can be quite corrosive to some plastics, so you have to ensure that your tubing and your bags are both going to be resistant, both in short-term and in long-term storage. You then have this overmolded assembly. This is something that Saint-Gobain is proprietary to Saint-Gobain, and it allows for the filling of small bags. And so in this picture, you see 2 ml bags. We can go down to 1 ml. and customize the outlet. So you can have a spike, you can have a lure, you can have a piece of tubing that could be sterile weldable. So these are all options that are available. In this example, they had a lure fitting. In the image here as well, so you can see how the fluid flows. And again, the shape of the bags, the freezing bags are more in circular form. And again, that's a preference of the customer and something that can be designed. Okay. With that, we're going to move on to the Argos case study. And I'd like to thank Argos for providing these slides to us. And this is really focused on their Acellis program. And essentially what their Acellis technology platform is really a truly individualized therapy. It's combining both the patient's cells and the patient's mRNA from their tumor in order to form a truly individual dendritic cell for an immunocell therapy for kidney cancer. So it is a platform, so it does have broad applications, but currently their phase 3 is in kidney cancer. It is ongoing, and some of the aspects that I'd like to talk about today are really the commercial model of it and the fact that Argos has decided to do automation or at least phase in automation for this indication and for this platform technology, and kind of how Saint-Gobain has been involved in this journey. So just to give a little bit more detail on the Acellis platform, it is a patented high-yield process, and again, it has two streams. So one is doing a leukapheresis on the patient, collecting the monocytes, and then combining it with an extraction from the tumor that allows the extraction of the mRNA, so getting a really nice signal from the patient's own tumor, and combining it with the monocytes so that they develop into very specific dendritic cells in culture. And from that one leukapheresis, they can get multiple doses for one patient, which minimizes the amount of times a patient would have to be leukapheresed. The automated manufacturing process. So right now, the clinical manufacturing is currently performed manually with open manipulations and biosafety cabinets using single-use disposables. But they initiated a custom equipment and single-use disposables project between Invetech, Argos, and Saint-Gobain to really look at automating the process to improve the consistency without having to have an expansion of the facility size or personnel requirements. And this is all done using closed single-use technologies, again, to mitigate that cross-contamination risk. So the automation equipment was designed by Invetech, and the closed disposables were provided by Saint-Gobain, and so this is how that partnership exists. This is a peek at one of the disposables, and again, this is phased in after commercial launch, but essentially this is all happening in a single facility, a scalable production process to meet the growing demand, and it is all automated with a hope that it could further lower cost of goods. So this is an example of the RNA manufacturing disposable. So I talked about those two arms, one doing the leukapheresis for the monocytes and the other for the RNA extraction. So this would be on that latter pathway for the RNA pathway. And this is a nice one to talk about because this is essentially no human contact. The equipment itself is never in contact with the subject material, and there's no human contact. It is all automated. And the reason for this, as I'm sure many of you could draw the conclusion of, is RNA is all around us. And so the idea here is that we need to minimize any contamination of foreign RNA into the process so that the patient cells are only activated with the RNA from their tumor. So here you see that there's a flexible barrier for the robot arm, there's the disposable vacuum pump head, there's the sealed tubing for aliquots, the thermal cycler lid for PCR, and the pipette head attached to the robot arm. We can take a sneak peek inside as well. So this is the insides, what it looks like. So you have the cuvettes, the elution pipette tip waste, vacuum waste, input and output wells, the PCR plate, reagent rack. So everything you could think of in terms of RNA amplification is within this disposable and is all automated. And you can think of the complexity that goes into a design like this and trying to understand what's required. So first of all, it requires a manufacturing facility and a process to have very stringent cleanliness requirements in order to avoid any contaminating RNA or even removing any of the RNA from the patient. Additionally, the material selection is of the utmost importance in these complex of a design, alongside being able to do 3D design. And this is important in the development because you need to choose the right materials up front. You need to ensure that they all work together and that they can all go through the different process steps, including the automation, the sterilization, and the process steps that lead up to being able to deliver a final product. The take-home messages on custom disposables. They can, and Saint-Gobain may argue that they should be an integral part of process development. So they can save on labor and materials, they can connect equipment and work cells. They don't necessarily need to be standalone solutions, but they could be things that fit and connect to unit operations and allow the close up between those unit operations. They can increase yield and efficiency, and overall they can improve patient safety and CGMP compliance by offering closed and less risk for contamination. They require specific expertise in terms of developing. So material science, selection, and processing, as I talked about before, is critical, especially for something like cell therapy where you have the interaction of biology and material science. Understanding that interaction is critical to ensure that the material is going to perform to the customer's expectations. 2D and 3D design is also critical in order for the customer to envision what it looks like and also to ensure all the pieces fit together. Systems integration, so this is looking at not just designing one item or two items, but several items and being able to understand how to put them together. The Argos project is an excellent example of this, but not the only one that Saint-Gobain has encountered. Quality systems, so with all the design and material expertise, you also need to have a robust quality and regulatory team that understands the different requirements from the customer standpoint and how to support beyond just delivering a product. And of course, the willingness and ability to customize. So not knowing that cell therapy is in a space where different customers have very different requirements, even looking at the autologous or allogeneic space, so those two spaces are very different, but also looking within those spaces and understanding the different cells people are using, as well as the different processes that those cells are taking. So we at Saint-Gobain understand that there is not an off-the-shelf solution that's typically applicable to more than one patient. And so the ability and willingness to customize allows us to create solutions that are specific to our customers' needs. And thinking about the manufacturing end game early. So we all understand that doing automation and full-blown closed systems in a preclinical or phase 1 may not be appropriate from a cost and effectiveness standpoint. But thinking about and envisioning how that system may look when you get to phase 2, phase 3, and commercialization, we believe is important, and it's something that we'd love to be involved in the conversations to understand what are some actions that we can take early on that will allow for that to happen later, as the product moves through the clinical trials. That's the end of my portion of the talk, and I think we have a few more poll questions. I'll let Elena take back over, and then we'll have the Q&A session to start. And I welcome any and all questions around anything that I've talked about or anything I may not have talked about that you're interested in. And thank you very much. I appreciate all of your time on this Tuesday morning.
Thank you very much for that insightful presentation, Natasha. So I hope our audience found it of great interest and value. So if you do have any questions for Natasha, as she said, then please do continue to submit them using the Q&A tool on the right-hand side of your screen. So before we begin the Q&A session, we have some more poll questions that we would like you to answer. So again, for each poll, please check the option or options that apply to you, and then just click the submit button. So the first question is, do you use single-use systems in your manufacturing? Yes or no? Thank you. The second question is, do you feel there is a need for custom single-use solutions in order to optimize your manufacturing process? Yes or no? Thanks very much. Then we have, which do you think is more likely the future of cell therapy manufacturing? Centralized model, decentralized model, or point-of-care manufacturing, or other? And if you could please comment, that would be great. Thank you. And lastly, what do you think is the largest gap in being able to manufacture cell therapies today? That's an open answer, so if you could please comment below the question, that would be great. Great, that was the last poll question. Thank you for taking the time to answer them. We will now move on straight to the Q&A section of this webinar as we have received a number of questions, and we will address some of these now live on air. So, Natasha, to get us started, where are your products manufactured?
Thank you, Elena. So, as I pointed out earlier in the presentation, we do have global manufacturing sites. Specific to cell therapy, our site is in Gaithersburg, Maryland, which actually has a grand opening next week on October 18th. And this is a brand new facility that is about 30,000 square feet of real estate and offers capabilities with ISO 7 clean rooms, ISO 8 clean rooms, and even ISO 5 assembly and packaging stations for very critical components. So we're very excited to see this new facility come to fruition, and I think the capabilities that it brings will be very exciting for our ability to manufacture single-use systems for the cell therapy market.
Thank you very much. So the second question we have here is, what sort of validation can you provide with your products?
Great question. So validation, of course, as I talked about, the quality and regulatory support is just as critical potentially as the product delivery itself. Saint-Gobain prides itself in a very robust quality and regulatory team as well as systems. In terms of the validation that we can provide with our products, we have a full validation guide on our fluoropolymer bags. And that goes from everything from USP testing as well as leachables and extractables that we can provide. We do have leachables and extractables capabilities in-house through our SGRS, through our Shanghai R&D facility. So this allows us to do some really nice profiling of our products and really understand the leachables and extractables profile of them. So that information is available to our customers.
Oh, thanks, Natasha. So what volumes are typically required in order to have a custom solution created?
This is also a great question. So typically when people hear custom, they get scared because they think it's a pretty large investment, or they think in terms of money or even volume. So people think of in thousands. And this is typically because when you're making, say, a bag, you typically need to invest in molds in order to do the hot bar sealing process. Now, what we do at Saint-Gobain for our bags is we have a laser welding process, which is proprietary. And this allows us to create very unique shapes, as some of the ones I've shown you in the previous case studies, by just programming the laser. This also allows us to make very easy changes. So through that process I discussed earlier, where we're kind of designing the custom solution, where we like to provide a sample and say that the customer likes everything, but they want the port changed, or they want the port location changed. So this is very easy for us to do. We can reprogram the laser in order to achieve that. And I think that is what allows our that kind of drives our willingness and ability to customize, because we don't need to have these large investments in tooling in order to provide these custom solutions. So typically minimum order quantities for custom bags and assemblies would be on the order of 10.
Okay, thank you. So another question here asks about the extractables and leachables profile of your disposable bags for cell therapy applications.
Yes. So the bags, as I alluded to in our validation, is we do have a full extractables profile on our bags available through our validation guide. So to whoever asked the question, if you'd like to reach out to me directly, I am happy to provide it. It is also available on our website as long as you're a registered user. So you can register and download the validation guide right off the website.
Thanks very much. So the next question here is, can you make assemblies without any animal-derived components, and also with all the tubing, etc., that is free from latex and is class 6?
Yes. So again, this is where the conversations with our customers come in. So if the specification is for ADCF materials, then that is something that we will specify into the design. We have access to industry-leading tubing formulations through our Saint-Gobain group. We have a multitude of connectors, of filters, and of bag materials. And specifically our fluoropolymer bag materials are ADCF. So the core that we're working with is ADCF. And again, our materials, if ADCF is a requirement, then that is certainly something we work with our customers to develop a solution that meets that expectation.
Okay, thank you very much. The next question is, what is the lowest temperature your product line can withstand?
So for this, I'm going to zoom in on our FEP bags, because this is typically where the cryopreservation is actually happening is with our FEP bags, and they're what's used typically for cryopreservation. And so the really nice thing about FEP is that it's a fully fluorinated fluoropolymer. So inherently by its chemical structure, its operating temperature can go down to below -200 degrees. So unlike other plastics that may have a glass transition point that is potentially at -40 or -50, and so below those points it's going to start potentially getting brittle. FEP doesn't have that, at least not before -196 C, which is the liquid phase of liquid nitrogen. Yes, can you hear me?
Okay, it seems that we've lost connection with our presenter. Please bear with us while we
try
to work this out. Thank you.
Can you hear me? Hello?
Hi, Natasha, are you there?
Yes.
Sorry, we just lost you for a second. Go ahead, please,
okay.
If you answer the question.
All right, excellent. So I'm not sure where I cut off, but focusing on FEP bags, so as I said, they can go down to -205 C. So they're going to remain flexible at that liquid phase in the liquid nitrogen, which is down to -196. So you're not going to have that brittleness that you might experience with other plastics. They don't really need additional mechanical support that you might need to use with other cryopreservation containers. So FEP is a great material for low temperature applications because of its inherent chemical structure. So I apologize if I got cut off in the middle of that. So if there's anything I need to explain more on that, please feel free to re-ask the question.
Oh, no worries, Natasha. I think it was just not us hearing us, but you, but the rest could hear you, so it's all fine. Thank you very much. I'm just going to follow with the next question. So are some of the aspects of custom design, so for example, bag shape for filling or draining, going to be commercially available for all? So for example, is improvements to some current off-the-shelf disposable currently available?
I'm sorry, Elena, can you repeat the first part of that question?
Sure, sorry, it's a bit of a long one. So basically, the question is, are some of the aspects of custom designs, whether they're going to be commercially available for everyone?
Yes. So again, this is certainly a case-by-case scenario, but typically, like the ones I showed today, there are often designs that are applicable to other customers. So say the sheath bag design, that was designed for the influx flow cytometer. I'm sure there are a lot of customers that are using the influx. So that's not a design that's specific and will only be available for one customer. So there are certainly designs in that case that may be more widespread. The point of my last comment in the conclusion is that often, even with kind of the one design, there's certain tweaks that may have to happen from customer to customer, and that's where we take the ability to kind of do that. But if one solution works for one customer and it can work for the other, then we absolutely would offer that.
Great, thanks very much. So another question we have here is, what is the state of technologies for injectable biodegradable beads?
So that is not my area of expertise in terms of biodegradable beads. Saint-Gobain does not currently manufacture any beads. So the bead example that I gave was specific to helping our customers separate out magnetic beads, but I can't comment on the industry for biodegradable because that's not an industry we're in.
Thanks very much. So the next question is, do you think an all-in-one automated system, blood and product out, versus automated unit operations in series is more practical, and why?
This is a great question, and I think one that a lot of the industry is trying to answer. So this is essentially the decentralized versus centralized model debate. And I think there's, not being in the cell therapy industry, so Saint-Gobain is a supplier, so coming from that side of it, I think there are definitely arguments for both. I think at this point, having an all-in-one automated system, so having the decentralized model would be very difficult. Not to say that it would be impossible, but it would be very difficult at this point with the technologies available. There's certainly been technologies that have come out that have tried to head in this direction, but based on kind of my understanding of what we're hearing in the industry, we're not quite there yet. And I think based on just the variety of cell types, again, and different processes that customers are using, I think for at least the foreseeable future, it does seem that kind of this unit operation approach may be what is more practical, again, in the near term. I think everyone has a different process that they're working with, and so the first step is the ability to at least connect those processes and potentially automate them. So looking at what Argos is doing and what others are doing in terms of trying to close up the system, trying to get rid of manual labor and get rid of open systems. I think that's kind of the first hurdle that the industry needs to get over. Is the decentralized model, the all-in-one system, a potential for the future? Absolutely. I think it's still on the table. I don't think that there's any reason to believe that that wouldn't be a potential solution moving forward. But I think at this point, a lot of people are focused on at least closing up the process and trying to get that cost of goods down in order to be able to have a commercial viable product. And from there, we'll see what happens. I think everyone is very excited to see how this market moves forward, and everyone, including Saint-Gobain, is here to help our customers do that.
That's great stuff. Thanks, Natasha. So how do you ensure that splitting is done in the same proportions, and what's the variability in volume that you get between the 10 different bags?
Okay, so I'm assuming this is in regards to the final fill and cryopreservation example. So great question. So this is where certainly we would need to work with you to understand what the tolerances of each of the filling, each of the volumes for the bags would need to be, and understand if there's any ancillary equipment that needs to be provided, whether it be pumps. So this is definitely something that would need to be engineered specific to the customer's requirements, and to understand again what their tolerances are. It'd be something we'd absolutely love to have a conversation with you about.
Great, thank you very much. Now I think that was the last question for today. So thank you very much, everyone, for submitting them. And of course, thank you very much, Natasha, for answering those questions for us today. So before we wrap up, do you have any last comments you would like to add? No,
I appreciate everyone's questions and attention. I'm happy that we got through all of this in under an hour. I know everyone has quite busy schedules. And so if you have any other questions, anyone on the line, please feel free to reach out to me directly. Or if for some reason I didn't answer the question completely enough, please reach out to me and we can have a conversation offline. And we'd be happy if you were interested in any of the cases that we showed today or in any opportunity to develop a custom solution with Saint-Gobain or even just learn more about Saint-Gobain's product offering, we'd be happy to have a conversation.
Thanks very much, Natasha. So I would like to once again thank you, wonderful presenter Natasha Lundgren, as well as our listeners, of course, for your time and questions. If you do have a minute, we would be grateful if you could complete our feedback survey, which you can find in the resource section of the screen. Please remember that you can find a recording of this webinar available to view on demand on RegMedNet at www.regmednet.com/channels/196-webinars, where you can also find details of other upcoming and existing webinars from RegMedNet. So thanks again for attending, everyone. Have a great day, and goodbye.